You recover. The fever fades, blood smears turn negative, and the diagnosis of malaria is considered resolved. Then, months later, it returns. No new mosquito bite, no new exposure, just the same parasite re-emerging from within. This kind of relapse is driven by hypnocytes, a dormant liver stage unique to specific species like Plasmodium vivax and Plasmodium ovale. After a mosquito injects sporocytes into the bloodstream, they rapidly migrate to the liver and invade hepatocytes. Most begin a process called exoerythrocytic skyogamy, replicating into thousands of merozytes that later infect red blood cells. But hypnocytes diverge at this exact point. At this point, instead of replicating, they enter a state of cell cycle arrest, DNA replication halts, metabolic activity drops to minimal levels, and the parasite persists as a small uninucleate form within the hepatocyte. Unlike active liver stages, hypnocytes produce very low levels of antigen, limiting detection by their host immune system. At the molecular level, this dormancy is associated with downregulation of genes involved in replication and metabolism, altered mitochondrial activity, and changes in epigenetic regulation. These changes stabilize a quiescent state similar in concepts to dormancy seen in stem cells or bacterial persisters. The reactivation process, though not fully understood yet, appears to involve the reversal of this suppressed state. The parasite resumes transcriptional activity, re-enters the cell cycle, and undergoes rapid schizogamy. Within days, thousands of new merozytes are released into the bloodstream, initiating a new erythrocytic cycle and clinical relapse. The triggers for this seem to be things like host immune changes, physiological stress, and intrinsic parasite timing mechanisms. But as of yet, no single mechanism has been definitively proven. From a clinical perspective, hypnocytes are significant because they are insensitive to most antimalarial drugs, which target actively dividing cells. Effective curing of hypnocytes requires drugs such as primaquine or tafenoquine. However, their use is limited by risks in individuals with glucose-6-phosphate dehydrogenase deficiencies, complicating widespread treatment. From an evolutionary perspective, hypnocytes function as a temporal reservoir. By delaying their development, the parasite bridges gaps in mosquito availability, synchronizing relapse with future transmission opportunities. This strategy increases long-term fitness without requiring continuous infection. Hypnocytes redefine infection as a continuous process, but as a discontinuous one, interrupted, hidden, and then resumed. The parasite does not need to persist in the blood to survive, it only needs to remain unseen, and when conditions align, it begins again, exactly where it left off. If you like this content, guys, please like and subscribe, and I'll see you in the next one.